Good day, grade 11 learners. Welcome to the Life Sciences lesson. My name is Shabnam Sukhnandan. This lesson is brought to you by Sci-Bono Discovery Center in collaboration with the Gauteng Department of Education. So learners, today we're going to also look at another chapter of photosynthesis relating to the greenhouse as well as the investigations regarding photosynthesis, which is actually very important because it deals with skills and how to apply your knowledge to the practical investigations, which is very important for grade 12 as well. So now, learners, we're going to look at what we call a greenhouse. Now, with a greenhouse, remember, do not get confused between the greenhouse effect and the greenhouse. Okay, the greenhouse, so you have something called a greenhouse, then you have the greenhouse effect, and you also get greenhouse gases. Now they are all different. So what you're looking at in front of you is actually a greenhouse that is actually a house-like structure where the roof is made with translucent plastic, right? It's a structure with a transparent roof for light to come through, transparent walls that go all around like a dome, and it's used to grow plants, and all plants are green. So because the roof is translucent, means light can come through, it's transparent, not so transparent as in clear, but translucent light must come through it. Okay, and you find the plants are growing inside, and because all the plants are inside, it's called the greenhouse. Okay, so that's the greenhouse what you are looking at in front of you. So learners, while we're busy on the slide here, what you need to know also is what is the greenhouse effect. Now a greenhouse effect is whereby, within this greenhouse that you are looking at inside, it's very, very warm. So even in winter, a greenhouse keeps the plants at an optimum temperature of 25 degrees Celsius, even if there is frost and snow outside. So it's very, very warm inside this house. And just like you sit in a car on a winter day, you find you'll enjoy being in a car when it's very, very cold. Instead of sitting in the house with the heater, if you sit in the car, the light of the sun comes through the glass, right? It comes through the glass, and then it traps the heat within the car and makes you feel warm. So same thing, all the light can come through the translucent walls and roofs, and then the heat is trapped inside. And when the heat is trapped within this house, it's called the greenhouse effect. And that is what happens when you talk about global warming. When there's too much of carbon dioxide in the atmosphere. Do you remember you did that in grade 10, right? When there's too much of carbon dioxide in the atmosphere, here's the earth. Okay, here's the earth. And there's too much of carbon dioxide that's accumulating below the atmosphere. That carbon dioxide acts like a blanket. It keeps the sun rays or heat within the earth's surface, within the atmosphere, and warms up the earth so much. That is why it's called global warming. So the greenhouse effect simply means that the temperature within that house is increasing, okay, because of the sun's rays being trapped. So that's a greenhouse where plants are kept. And then you have a greenhouse effect, which is where heat is actually retained and prevented from escaping. And then you have what we call the greenhouse gases. And the greenhouse gases are what is actually going to contribute to global warming. Okay, so you find global warming is when the Earth's temperature is increasing too much and trapping the heat, right? But now what's causing it to be trapped is not only carbon dioxide, but other gases like methane, sulfur dioxide, nitrous oxide. All those are methane, those gases are actually greenhouse gases, and that causes the sun's heat to be trapped within the earth. So there's greenhouse gases like your CO2 that come from the burning of too many fires. Then you have the greenhouse effect whereby within the house it's warm, very warm, and your greenhouse is a structure looking like a house made with transparent walls and transparent roofs with plastic, so that light can come from the sun and get trapped. The heat of the sun gets trapped within that house, and here your plants are growing, so that you can get lettuce throughout the year. You can get beans throughout the year because it's actually grown in a greenhouse. So irrespective of outside temperatures, these plants are going to grow. And how do they grow? By the process called photosynthesis. Right, so learners, here you can see greenhouses can be used to maintain the optimum level of the factors affecting the rate of photosynthesis. So what are the factors that affect photosynthesis? Remember, we did that last week. It was CO2, yes, and it was water, it was light, right? It was temperature as well. In other words, a plant cannot photosynthesize without CO2, without water from the soil, without light from the sun, and obviously the temperature has to be correct. For the enzymes that control photosynthesis, they need to function at the optimum, the most suitable temperature for photosynthesis, which is 25 degrees Celsius. So all the factors that we mentioned now, the CO2, the water, the temperature, and the light, have to be at its optimum. If you give a plant too much of CO2, you can increase the level of photosynthesis to a certain extent, but then it won't go more because all other factors have to also be increased. So if you're only giving one factor, increasing one factor, then the other factors will become limited. You call them limiting factors now, because you're only increasing one and the others are the same. So if you increase one, you've got to also increase the rest. Okay, but to a certain level. So light passes through the transparent structure, or if there's no light, if it's very, very dim, it's a very cloudy day, then you find within the greenhouse, the farmers actually put artificial light sources, like certain kinds of lamps, which actually increase the light intensity within the greenhouse and allow the plants to photosynthesize for a longer time. So especially in winter when the days are shorter, so the farmers will use certain lamps, right? These fluorescent lamps which give off a lot of light, so that the light burns longer, so that photosynthesis can occur at least by till 8 p.m. So there's more photosynthesis, more production of plant and food crops. Okay, so carbon dioxide is also present in the atmosphere. So within this greenhouse, you're getting light, you're getting the right temperature because the heat is trapped from the sun, but you also must give the plants what? CO2. So now you've sealed off these plants. So what can you do to increase the amount of CO2? CO2 can be pumped into the greenhouse, right, using a little like a hose pipe. Then they pump out a gas called CO2. It also comes from certain kind of lamps that can burn, and CO2 is given off. Or they can also put a lot of sodium bicarbonate in the soil, and then you find that the sodium bicarbonate will release CO2 into the atmosphere within the greenhouse. Okay. The temperature can be kept at optimum using heat and cooling devices. So the greenhouse, when it's very, very hot in summer, then the farmers use cooling devices so that the temperature does not exceed 25 degrees. And when it's very, very cold, then they will use lamps, whereby the lamps will give like light, right? And they also give off heat. So that means the temperature must be correct, the CO2 must be correct, the light must be correct, and obviously the plants must grow in a well-watered soil, but not too much of water. Right, so let's go again. How do the greenhouse gases work, or how does the greenhouse work? You can see some of them occur in the form of a dome, and some of them are very small, like a little house. So they have to have struts, and over the struts they put this like a translucent plastic, but not just on any kind of a plastic. It has to be like a polyurethane plastic. Okay, you get, the polycarbon plastics, but the clear ones that you can sometimes use in your sky roofs. Yes, that's quite strong, and that wouldn't actually break with the hailstones and wouldn't just fly away in strong winds. So they have to have a special texture for the greenhouse. So it cannot be flimsy, it must be very durable, and it must withstand the weather conditions and also protect the plants. Okay, so here's your greenhouse, and you can see here there's a whole lot of potato plants, and here you can see there's more lettuce and things growing in this greenhouse. So light comes in order to provide light. The greenhouses need to have some way for the light to come in, and that is by the translucent plastic. This is why greenhouses are made up of translucent materials like glass or clear plastic. And remember, it's not the clear plastic that you use in your like plastics whereby you can put in crisp and things like that. It has to be hard, durable plastic. But translucent means light must go through, so it mustn't have a color. So this gives plants inside the maximum access to sunlight. So very important. Here you can see a little sketch of the sun and the sun rays coming through, going through the glass, translucent glass, and then going in there. And obviously this door will be closed as well, or you can even leave it slightly open for what? For carbon dioxide to enter, for oxygen to exit. So you can leave it slightly ajar, but obviously there mustn't be too much of wind, and there cannot be even animals that can enter. So there has to be some control system of this door. It cannot just be left open all the time throughout the night and day. But anyway, this door is open here to allow some gases to enter and leave, right? And you can see that as the light rays are going through the glass, these little arrows in red are what? Those are infrared light. Okay, so that means light rays has a different wavelength compared to the wavelength of heat. So wavelength of heat is different, and because it's slower, it gets trapped within this greenhouse. So you see, when the light comes into the glass walls of the greenhouse, it is absorbed by the plants because chlorophyll. What is plants having? They have chlorophyll, and the chlorophyll is the one which is going to be energized by the light energy of the sun. Do you remember? You did the light phase, right? So in the light phase, it's the chlorophyll that's going to trap the light, energize the chlorophyll molecules, right? And that's to start off photosynthesis. Then the greenhouse is also absorbed by the plants, by the ground, and anything else in the greenhouse. So the light gets absorbed, converting it into infrared energy. And what is infrared? It is actually heat. So heat energy and light energy are two different things. Okay? A heater gives off light, and it also gives off heat. Even a lamp, it will give off light, and if it's on for a very long time and you touch the bulb, it's very, very hot. So each of those are energies. So light energy has a different wavelength, and heat energy has a different wavelength. Right? It's like sitting in the car in winter. Light comes through the window and the heat is trapped. Okay. So here you can see your plants. The soil is giving off infrared radiation because even the soil absorbs the heat of the sun. Right. And you can see all the light rays are coming in here, and it's going to be contained in this greenhouse, just like this. And here's the heat going up, and it's circulating and keeping the greenhouse at an optimum temperature of 25 degrees Celsius. What else do you think that the farmer must have installed in his greenhouse to ensure his temperature is always correct? Good. He must have a thermometer, because remember, you must keep the temperature within the greenhouse at its optimum, not too low, not too high, but the most suitable. So if the mercury reading in the thermometer goes very, very high, then they've got to open the door or use cooling devices to bring the temperature from maybe 32 to 25. Okay. And if it's very cold outside, like you have this cold front, it's snowing, it's very, very cold, then obviously they cannot just put heaters, but they got to use some kind of lamps that are giving off heat, but at least till 25 degrees Celsius. So thermometer needs to be there. A huge thermometer needs to be shown in the greenhouse, and obviously they must have people to supervise the greenhouse to ensure that their soil is well watered, that the doors are closed at a certain time, that there is enough sodium bicarbonate in the soil to give off CO2, the plants are well watered, that there's not too much of dust accumulating on the walls of the greenhouse, right, on the roofs of the greenhouse, because too much of dust will block the sunlight. So they always need to be ensure that the glass they use can be rinsed off easily and allow light to come through. Okay. So now the same diagram here, you can see here's the sun rays, and you can see these wavy lines indicate the wavelengths of light. Okay. And here you can see now the sun's short waves of light, and the infrared rays radiate from the ground and cannot pass through the glass. So the heat waves cannot pass through the glass. So the long wavelengths radiate to the atmosphere. So the longer wavelengths go back to the atmosphere, and the shorter wavelengths of heat stays within the greenhouse. So you can see here that the blue is indicating shorter wavelengths of sunlight, and then the longer wavelengths regards heat. So heat has a longer wavelength and light has a shorter wavelength. And because they work on different wavelengths, one is able to heat and one is able to give off light. Okay. So once the light energy gets converted into infrared energy, remember learners that energy cannot be created or destroyed, but changed from one form to another. Do you remember you did this in grade 9? What do we call that law? It's called the law of conservation of energy, which we apply in photosynthesis. So actually the light of the sun that's going to be energizing the chlorophyll molecules will be converted in the chloroplast now into chemical potential energy, which is going to be stored where? In the products of photosynthesis called glucose. Remember, the main purpose of absorbing light is to activate the chlorophyll molecules. And once that energy is there in the chlorophyll molecules, it's going to now help to actually produce glucose, and that energy of the sun is going to be trapped in the food. And that is why when we eat food, that energy of the sun transferred to the food as chemical potential. Then when we eat it, the chemical potential is converted to kinetic energy. And that's the energy that we use to do work. All right. So it all starts off from the sun. Do you remember in grade 10 you did the food chains, and you always start off that the most important abiotic factor is the sun, and from the sun the energy moves to the plants, and then from one organism to the next, right? So the change in wavelength makes or prevents the heat from escaping the greenhouse walls. So the differences in wavelength prevents the heat from escaping because it's a long wavelength. So by the time it moves, it's going to take a long time to escape. Therefore, it gets trapped in the greenhouse. The air inside becomes warmer and the temperature will rise. Greenhouse effect. Light comes through and the heat will be retained, keeping the house warmer in winter. So you can even get roses. Mother's Day is coming up and you're going to buy your mom roses. We are getting close to winter, but you get beautiful flowers. So you find in City Deep Market, there's plenty of greenhouses whereby they are growing roses and all the different kinds of plants and all the different kinds of vegetables and fruit and like lettuce, okay, and your spinach. You find, how do we get them in winter? Because they are grown in the greenhouse. And because they are specially grown, they will be a bit more expensive. So flowers are more expensive in winter because they have been specifically grown in the greenhouse. Okay. So promoting photosynthesis. That's what greenhouses do. So you can see a whole lot of flowers or plants here, different kinds of pot plants with different kinds of flowers and little shrubs here as well, right? And they are all growing in this greenhouse. You can see how the panels are there to actually hold the translucent glass in its place. Can you see? And the glasses are quite clean. You can even see the sky. I told you because it has to be kept very, very clean so that light is not blocked out. So all this light and warm temperatures give plants ample access to sunlight and temperatures they need for growth. Remember, the optimum temperature for plants to photosynthesize is 25 degrees Celsius. So here's your greenhouse. Now we're going to have a look at an activity. A tomato farmer was able to produce crops in a greenhouse which were heated between 15 degrees Celsius and 22 degrees Celsius. So in addition, carbon dioxide level was increased from normal 0.03% to 0.1%. Now, look at this greenhouse. What is this activity dealing with? It's saying that in this greenhouse, we need to produce tomatoes. And that farmer has a greenhouse. So at what temperature is he keeping the greenhouse? He's keeping it between 15 degrees and 22 degrees Celsius because the farmer has studied that that's the best temperature in which tomatoes can grow. So he has to monitor the temperature within that greenhouse. In addition, the carbon dioxide level was increased from normal 0.03 to 0.1. Now children, remember, you may think that how can you increase something from 3 to 1? Remember, 0.03 is less than 0.1. This means it's 3 out of 100. And this means it's 1 out of 10. Can you see? 3 out of 100 is it's like 3 amongst 100 is too few. But 1 amongst 10 is not so few. It looks, you can pick out the 1. But 3 amongst 100 people is very difficult. So 1% is actually, or 1 over 10, okay, is a greater amount of CO2 level compared to 3 out of 100. So the farmer said, let me see if I increase my carbon dioxide concentration to from 0.03 to 0.1. And his temperature was actually between 15 and 22. So the fact that he could control his temperature, it means temperature was his controlled variable. So children, it's very important that you learn when you're doing experiments or investigation to know certain factors that you can control and keep the same. It contributes to something called validity, because you're keeping the temperature at the same level to ensure that the results are fair, so that you're not giving the plant more than 40 degrees or less than, this is the best suitable temperature, I have to control it. So that contributes to what we call fairness and validity. That's what he's able to control. Now he's changed the carbon dioxide concentration from 0.03, which is the most suitable, and he increased it even further now. State one way in which the farmer could have enriched the air inside the greenhouse with carbon dioxide without burning any fuel. So what does this mean? Learners, do you remember what is fuel? What is the fuel here? It's not petrol. When we talk about fuel, we talk about wood, paper, anything that you can burn, okay, that is responsible for combustion, can burn. And what does burning do? It releases heat, fire, and CO2 as an invisible gas. So now we're saying, what method can the farmer use instead of making a fire and putting the greenhouse at risk? What can he do to increase the CO2 concentration within the greenhouse to increase the photosynthetic rate. He can use carbon dioxide, he can use sodium bicarbonate in the soil. So in that case, you're not using or burning anything. Remember, fire gives off CO2. We don't want any fuel, we don't want any burning without burning. So you can put sodium bicarbonate in the soil, and then that will release CO2 into the air. You can also get a carbon dioxide pump, right, where you get carbon dioxide from a pump, and then you can slowly pump it into the greenhouse using like a hose pipe. And that will actually increase your CO2 level without any fires. All right. So you can pump CO2 from tanks into the greenhouse. You can add sodium bicarbonate to the soil. Remember, sodium bicarbonate is Na, that is sodium, HCO3. That is sodium bicarbonate. Okay. Add organic material that can decompose to the soil, such as methane from animal dung. So learners, remember, when you add fertilizers or compost or manure to the soil, what is happening there? What is that? It's all organic material, all your dead leaves, your dead insects, the droppings from birds, the droppings from fish. If you collect all the droppings from cows, from donkeys, if you collect all that droppings and make it into a heap, and then seal that heap and have a little tube going from that heap, the gas that's going to be released from there, as that leaves decompose or get rotten, what gas will be released? It will be CO2. So you find any organic material that is decomposing, you can have your own compost heap. It will release CO2 into the soil, and then, or even from animal dung, and you can increase the CO2, right? So what are the two ways? There's three ways here. So you can say he can use CO2 from tanks and pump into the greenhouse. He can increase the CO2 concentration in the soil by adding sodium bicarbonate, right? And he can use decomposing organic material which releases CO2 into the greenhouse. And there was no fires involved. Okay, so the next one is explain how increasing the CO2 levels up to 0,1 can lead to an increased production of tomatoes. Now, how can increasing the CO2 increase the amount of tomatoes that are produced? Let's see. First of all, if you increase the level of CO2 from 0,03, remember the most suitable temperature is, the most suitable level for CO2 is 0,04%. But now you're increasing it from 0,03 to 0,1. Okay? So how does it cause more tomatoes to be produced from this one greenhouse? First of all, the increase in CO2 will cause the rate of photosynthesis to increase. Higher photosynthesis, more starch, more glucose, more fruits are produced. Tomato is regarded to be a fruit, right? More sugars or glucose produced converted into starch. So all that glucose or food will be converted into starch and stored into the tomatoes. But tomatoes don't have starch. They are normally sweeter, so they have like fructose or glucose that is stored. But it's a result of photosynthesis. And also, the more sugars the plant makes, the more the fruits will be able to be born from that plant. In other words, those plants can bear more tomatoes because they have enough energy to produce tomatoes. Remember, the plants must now produce the tomatoes from the ovary of the flower. Do you remember we did in grade earlier, we did the plants, and you know the female part was what? The stigma, the style, and the ovary, ne? And in the ovary you had all your seeds. So remember that this ovary now becomes your fruit, which is the tomato. But for this ovary to swell up and grow into a tomato, it must have energy. And where does energy come from? Glucose. And where does glucose get made? In the dark phase of photosynthesis. So increasing CO2 will increase glucose and more tomatoes will be produced. So the rate of photosynthesis will increase the production of tomatoes. More glucose is produced, which will be converted into starch. You can also get potato plants. You know, your up-to-date potatoes are also grown in the greenhouse. That's why they are very, very expensive. They are grown in certain conditions. So it gives more CO2, more water, more light. So more glucose is produced, more starch will be formed, and the bigger the potato, because potatoes are starch. So you're going to get very big potatoes. That is why an up-to-date potato is very, very big compared to the ordinary potatoes. And they cook faster as well. Okay, so you need glucose as energy for growth for the plant in order for you to produce fruits and crops. Right. Same question again. Same investigation, different questions. Name 2 other factors which might have increased to obtain an even greater yield when the CO2 level is increased to above 0.1. So what this question, you must analyze the question, learners. They are asking you, if I increase the CO2 level from 0.03 to 0.01, I'm increasing photosynthesis. I'm also increasing the amount of glucose produced by the plant, and that glucose is going to be used to produce more fruits. Right. Now, what else can I do? Can I only give the plant a lot of CO2? No, because CO2 is not the only factor for photosynthesis. What other factor must you ensure? If you're increasing CO2, you must also increase what? Light, good, and temperature. So you must increase the temperature, because if you only give it lots of CO2, then temperature and light are going to be what? Limiting factors. So CO2 will just increase, and then there's not enough light, it's not so hot, and then so photosynthesis will slow down. So if you want to increase photosynthesis, if you increase CO2, you must also ensure that your temperature and your CO2 is also increased. So you can't just increase one and leave the other 2 to be stable. They also have to be increased to a certain level. All right. Now predict what would happen if the CO2 concentration within the greenhouse is increased to 0.5 between 10 to 15 days if all other factors are kept constant. Now, if we're going to constantly increase CO2 from 0.03 to 0.1, we saw photosynthesis is going to increase. If it now goes to 0.5, photosynthesis will increase, but then it's going to slow down. So it's going to look like this in a graph. Photosynthesis will increase because you're giving it more CO2. It will increase, increase, increase. And then no matter if you go beyond 0.5, it will just flatten out like that. So this optimum here is going to be 0.5% CO2. But then the photosynthetic rate is not going to go on like that. It's going to come like this and stable out. Why? Because CO2 also is not the only factor. You need CO2, you need water, you need light, and you need the most suitable temperature. Okay, so let's just erase this here now. Okay, let us see the answer. If you increase CO2 from 0.5 from 0.03 to 0.5, which is 5 over 10, right? There will be an increase in the rate of photosynthesis, yes, for 10 to 15 days, because you're increasing it between 10 to 15. Obviously, you'll see photosynthesis will increase, right? But all other factors are kept constant. So after which the rate of photosynthesis will decrease as other factors become limiting. So the graph can go like this. It will increase, increase, increase. And then it can go down, and it can actually eventually decrease. Because now, here you're increasing your CO2. You're increasing it too much. Now water, light, and temperature become limiting factors. So photosynthesis will decrease and level out, and eventually all that CO2 will be there will be not enough water, and then you find photosynthesis is going to drop. Okay. So there will be an increase in the rate of photosynthesis for 10 to 15 days. You got to relate to the question that between 10 to 15 days at 0,5, the photosynthetic rate will increase to a certain optimum level, right? Thereafter, the photosynthetic rate will decrease as other factors become limiting. All the factors need to be at the best, the most suitable for the highest photosynthetic rate. In other words, all the ingredients or raw materials, if you increase one, you've got to also increase the dose of the other. Okay. So now, investigations in photosynthesis, right? There are investigations which can be performed to determine if a factor is required for photosynthesis. Now, there are many experiments that actually deal with photosynthesis, okay, to determine the rate at which photosynthesis is occurring. So now we must look at the experiments regarding the raw materials. You have an experiment for light. You can have an experiment, is CO2 really necessary? Is a photosynthetic really needing a certain temperature? Does photosynthesis really require chlorophyll? Okay, does photosynthesis really require light? So you can do an investigation to check, does photosynthesis require these raw materials? And learners, what you need to know is that when you are looking at these factors that are going to be investigated, the outcome is what? What is the purpose of testing whether these factors are there? To see whether starch or glucose is produced. If there's no glucose or starch in a plant, it means no photosynthesis took place. Okay. So in the investigation, one plant or one leaf, it's called the experiment, is given all of the requirements except for the factor that's being tested. So you want to test, for example, if light is necessary for photosynthesis. So you're going to have a pot plant here, and you got some leaves like this, okay. Here's your pot plant and your leaves. So what you can do is, you can actually take this plant now, right, and see, I've already have this plant, it's given everything. It's got water in the soil, it's got light from the sun, it's kept at the suitable temperature, so it's got everything. So obviously I can find starch in this plant. So when you're starting an experiment, learners, the most important thing that you need to know is that you have to remove that starch. You must remove the starch so that when you're doing the experiment or the investigation, you must know that the starch that's coming is from the investigation itself. It mustn't be there prior to the investigation. You must remove all starch before the investigation starts. So if you are testing for starch in a plant, that starch should come during the time when the investigation was being done. But we'll see later more about that, about destarching. So we want to know what is an experiment and what is the control. So I'm going to take a plant here. I'm going to now, I know it's got starch. I put it into a dark cupboard. I let all the starch be used up. Why dark cupboard? Dark cupboard means no light phase, no products of light phase, no dark phase. So no starch is made. And the plant, how is it going to survive? It will survive on all the starch and glucose that's already there. So when you place it in the dark cupboard, the plant won't die because it will use up the glucose and starch that was there already. So if you leave it for 48 hours and then you take it out now the plant got no starch or glucose. So you start your investigation. You're going to take a leaf and you're going to say, let's take this leaf here and put it into a test tube and seal it, or into a flask and seal that flask. So you'll have like a tripod stand here and it's holding the flask into its place. And in here, you put sodium bicarbonate. Say you put sodium bicarbonate, okay, which is a source of what? CO2. Now you're giving this plant CO2. Okay, you put sodium bicarbonate, you're giving it everything. So this becomes what we call a control. You see, another plant is given all the requirements in the same investigation and it's referred to as a control because it's got everything now. Now we take another flask, another conical flask, seal it here, put a tripod stand, hold it in place, hold the flask in place, and here we're going to put caustic soda. Now caustic soda is also referred to as sodium hydroxide, or you can use potassium hydroxide. Now what this does, it will absorb all the CO2 that is around these leaves. So you must ensure that no CO2 is entering this leaf from the side over here. It must be completely sealed. And that the only source of CO2 is sodium bicarbonate here. It's not coming from the outside and it's not escaping. And here, any CO2 that's around this leaf is going to be absorbed by the CO2. So sodium hydroxide absorbs CO2, whereas sodium bicarbonate releases CO2. Remember, learners, sodium bicarbonate, what does it do? It releases CO2. Sodium hydroxide, what does it do? It absorbs CO2. So it's going to deprive this leaf of CO2. Right. So you can leave it for a full 24 hours and let the investigation continue. Next day now, we want to see, was CO2 really necessary for photosynthesis? So I know this leaves on this side has everything. It's well watered. It's placed in light. It was placed in light for 24 hours. It was given light. It was right for a day, night. It was given light. There was light phase. There was dark phase. So, and it had CO2 as well. This one, remember, it had no CO2. So now you're going to do a starch test, okay, to test if starch was found here or if starch was found there. Remember that we had to place it, before we did this experiment, what did we do? We had to place it in a dark cupboard. What's the reason? Remove that starch before we start the experiment. What is the process called? De-starching. Now the experiment is there. The teacher will show you the plant. Let's take these leaves and put it in a conical flask. Let's take those leaves and put them in a conical flask. Let's give these leaves sodium bicarbonate. Let's give these CO2. That's the carbon caustic soda. Caustic soda absorbs the CO2. This plant here does not have CO2. It's going to be called the experiment. Okay, that one that's going to get, that's not going to receive the factor you're testing, it's going to be called the experiment. So in the investigation, one plant, the experiment is given all the requirements like light, heat, water, right? But only thing it's not giving the factor you're testing, which is CO2. Is that necessary? So that becomes my experiment. So in most investigations, a test for starch is performed at the end to prove that photosynthesis took place. You will need to know the steps of conducting a starch test. So you need to know two important things, learners. Before I do an experiment related or investigation relating to photosynthesis, I must de-starch the plant. And then I start my experiment from fresh to see if starch is formed. Why? Because when you determine if starch is formed, that's the way you're going to measure the rate of photosynthesis. Did photosynthesis take place? And how you measure how you're going to measure it? You remember, the rate of photosynthesis or the formation of starch becomes what we call a dependent variable. So the formation of glucose and starch depends on, or the rate of photosynthesis depends on the amount of the factor that you are testing. That depends on how much of CO2 you're giving, how much of light you're giving, how much is the temperature. Né? So all that will actually contribute to what? the rate of photosynthesis, and the higher the rate, the more the glucose, the more the starch. So that is the photosynthetic experiment. So let's go to the next one. Now here is the investigation one, the starch test. So learners, these are very important factors that you need to know. During photosynthesis, glucose is produced, which is converted into starch when glucose molecules join. So here is your glucose. Now, many, like let's say for example, in the 1980s, the metric syllabus used to have photosynthesis as part of it. And one very important thing that we needed to know those days was that how does glucose form starch? And you needed to know the structural formula of glucose, which is C6H12O6. For those that do physics, know the structural formula of glucose. And this is how glucose is actually looking like. It's a hexagonal shape with all your carbons and your oxygens and your hydrogens, which are in that ratio there, and they must join by removing water. So the H and the O and the H will join. The H and O from the one group joins with the H from the other group. Okay, your hydrogen joins with your hydroxyl group. They form water, and that when that water is removed, all the glucose molecules will join to form starch. So these little beads that you see here, this is not a necklace, but it's a starch grain. So a starch grain is made up of millions of glucose molecules joined by a process called dehydration synthesis. You don't need to know that now, but it's called dehydration synthesis because as the glucose molecules join, water is removed. And then the glucose is a simple structure. It's called a monomer, and they join and join and join like pages together to form a book. So the book becomes your starch and the pages becomes your glucose molecules. So the little units are called monomers, and the full composite unit or polymer is called your starch. So here is how starch is formed. To determine whether photosynthesis occurred, how are we going to know? Because it's abstract. You don't know if photosynthesis is occurring. You're saying, I'm giving my plant light. I'm giving it a suitable temperature. It's green. It has chlorophyll. I gave it carbon dioxide in the soil. It's getting enough light. But I don't really know whether photosynthesis is happening. So how can you measure that? You've got to do a starch test to check if photosynthesis is occurring. So if starch is present, then it can be concluded that photosynthesis has occurred. If starch is absent or not present, there's a negative starch test, then you can conclude that photosynthesis did not occur. Okay. Now we're going to do the starch test, and learners, you must do the starch test or your teacher has to do a demonstration of the starch test. Now here is this pot plant, okay, and you can see they're placing it in a drawer and they're going to put it into a dark cupboard. So this is the cupboard, and obviously here's the drawer of the cupboard. It's going to close. So we're going to do the starch test. What's the first step? De-starch a plant. So before starting the investigation, starch must be removed from the plant, okay, because it was already standing in the sunlight with all the relevant raw materials. So obviously you know that plant is photosynthesizing because it's growing, it's green, it's not dying. But now how do we know? We want to start fresh. So you take that plant that was well watered and healthy, you place it in a dark cupboard for 2 days, for 48 hours. So to do this, you place the plant in a dark cupboard for 48 hours, okay? We want to de-starch it. The plant will use the stored starch during the 48-hour period. What is it going to do? It's going to use up the starch that was already made when it was standing in light. So by using the starch up, we call it de-starching the plant. All right. It can be proved that the starch present at the end of the investigation is due to photosynthesis occurring. So That's the only way. We must remove all the starch. We can, even before we start the experiment, take the leaf and do the starch test. And you know what is the chemical we must use for starch test? We must use something called iodine, which is naturally yellow-brown. Iodine, we use iodine to do a starch test. And the minute you take the plant out of the dark cupboard, test for starch in all the leaves, and it should be yellow-brown because it's no starch. If starch is present, that iodine, which is yellow-brown, will turn blue-black. And when it turns blue-black, you know starch is present. You'll say, this plant is not yet de-starched. I'm still getting starch. So what you must do, you have to keep it for further another 48 hours until all the starch is definitely used up. Then you can test one leaf. No, this one is showing yellow-brown. I've removed all the starch. Now let me start fresh with my experiment, okay? So you're going to start. Now, how do you go about doing the starch test? So now we're not doing the experiment. We want to know how to do the starch test. So you want to see now if starch is present, okay, after an experiment, or you want to check if starch is present after you've de-starched. You want to make sure that there's no starch before you do your experiment. So how do you do the starch test? This is very, very, very important, learners. You need to know the steps. So what is the method? You need to know the method. First of all, you take a leaf, place a leaf in a beaker of boiling water. So what do we call this here? You need to know that this here is what we call a tripod stand. It looks like that, okay? It's a tripod stand, and your teacher should have it in your laboratory. It should be somewhere there in the laboratory, the tripod stand, right? Okay, let's just erase this and draw it. So here is your tripod stand looking like this with 3 legs like that, okay? And there's like a wire net. You can see it's like a net over here, like a mesh of wire. Then you're going to take a nice beaker of water and you're going to place it on there, and there's your water. There's your water. Now you're going to take a leaf. Okay, let's go here. Okay. You're going to take a leaf. Okay, let's just erase this here, learners, 1 minute. You're going to take a leaf from the plant you de-starched, okay? And you're going to put it into this boiling water. Now, when the water boils, it's going to kill the cells of the leaf. It's going to kill the cells. So place a leaf in the boiling water. This makes the leaf soft and kills the cells, because you want them to be dead now, but you don't want them to metabolize. You're killing them with the hot water. Now, once you've actually boiled this leaf and it's very, very soft, you're going to take this leaf now, which was boiled in the water, and you're going to put it into a test tube. Right, you're going to put it into a test tube like this. You're going to put that leaf that's soft into the test tube. Okay. Then you're going to fill that test tube with what we call alcohol or ethanol, which is found in the laboratory at your school. You fill the test tube with ethanol until it covers the leaf, like that. Don't fill it too much because ethanol is flammable. So it must just be enough to cover the leaf. Right. Then you take this test tube using a test tube holder, which is like a little metal piece with a handle, wooden handle. You take that test tube with the test tube holder and you insert it into the beaker of water like that. Can you see that? You boil the leaf, you kill the cells, you took the leaf, you put it in the test tube, you fill the test tube with ethanol, right, which is alcohol, not too much, just until the leaf is immersed and above the leaf. And you're going to put that test tube with the leaf into the boiling water. Now, what you need to know, learners, is that ethanol or alcohol boils faster than water. So, alcohol or ethanol boils at 75 degrees Celsius, whereas water boils at 100 degrees Celsius. So when this water is boiling, you also need to put a thermometer into this beaker. The minute the temperature reaches 75, this ethanol will boil first before the water, because 75 degrees. So the ethanol is going to boil faster than the water. And as the ethanol boils, it will extract all the chlorophyll out of that leaf, because the cells have already been killed with hot water. So all the chlorophyll is going to be extracted. See, beaker of boiling water, test tube with the leaf in alcohol. Right, you've done that. Allow the test tube to stand in a beaker of boiling water for 10 minutes. So how long are you going to leave that there? You're going to leave it for 10 minutes until the temperature in this beaker reaches 75. When you see that, you'll see the ethanol is going to boil faster. Then later the water will boil at 100 degrees. As the ethanol is boiling, you're going to see that the clear ethanol is going to turn a beautiful green, and the leaf is going to turn white, because all the chlorophyll has been extracted. So allow the test tube to stand. Ethanol cannot be exposed to direct heat because it's highly flammable, as I've indicated, and it has a boiling temperature lower than water, which is 75 degrees Celsius. This is why it's placed in a water bath. So we cannot, there will be a question, why can a test tube of ethanol not be placed directly over an open heat? Remember here, what's missing here now is a flame. You're going to put a Bunsen burner. Here's a Bunsen burner, okay? So here's the flame. It's got a little lamp there with some methylated spirit, and that's called a Bunsen burner. So this Bunsen burner is lighting, and you cannot hold the test tube over the in other words, if this is your Bunsen burner, let's say like that, okay, and it's got a flame, and here's your methylated spirit, you cannot put your test tube with your ethanol and the leaf directly like that. That is wrong. It's going to crack the test tube, because ethanol boils faster and it can squirt into your face. So therefore, whenever you are using ethanol, what are some of the precautions you need to use in the laboratory or be aware of? That the test tube of ethanol should not face you. It should face the direction where there are no learners. Okay, so if all the learners are here and you are observing the experiment, that test tube should face the other way, because we don't want the ethanol to boil over and burn you. Okay, so here is the water boiling. Here's the test tube of alcohol, and remember, chlorophyll is soluble in alcohol. So chlorophyll is soluble in alcohol and will be extracted from the leaf. The leaf will turn white in color and become brittle. It becomes very, very hard. It's no more soft like when it came out of the boiling water. The alcohol makes the leaf very, very hard, like how you feel like it's going to crack. That's why it's called brittle. All right. Now, carefully remove the brittle leaf from the alcohol. So you're going to remove that leaf. We're using a forceps. You're going to take a forceps, like a tweezer, and you're going to pull that leaf out of the ethanol. It's going to be brittle and hard. And then you're going to rinse it into that beaker of water. Remember, your water beaker was boiling. You're going to take that leaf now, and here's your boiling water. You're going to rinse that leaf in the boiling water, and it's going to make it soft. Now, once you remove the leaf from the boiling water, you're going to put it into a small Petri dish, a little dish like that, a small plastic, clear plastic dish, a very small, like a little saucer, but it's clear plastic. It's called a Petri dish. You need to know all these, learners, because when you go to varsity, you'll have to do all these experiments, and you'll need to know what's a Petri dish, okay? You need to know what's a Bunsen burner. You need to know what's a tripod stand. You need to know what is a wire mesh to put over the tripod stand in order for the beaker to sit, so that you don't get direct heat and crack the beakers, which are made of glass. So now you're going to take that leaf, which is now softened in the hot water, and you're going to put it into that. You're going to put it into your Petri dish, okay? Then you're going to take iodine, which is a chemical reagent, it's yellow brown. You're going to take the iodine and you're going to place it on the leaf. You can place it everywhere. You can put a drop there, you can put a drop there like this, you can put there, there, there. And then if the leaf is yellow brown or turns yellow brown, it means that the color of iodine is there and that there is no starch. So no photosynthesis took place. If the color changes to blue-black like this one, then you know iodine turns the leaf blue-black. Then you know that starch is present, because it's actually the starch. When it gets the iodine color, it will just convert the iodine into blue-black, which is an indicator. We call these solution indicators. It's indicating that starch is present when it turns blue-black. And this is called a starch test. Okay, so every time you do an experiment, you need to do the starch test. When the leaf, then what is the results? When the leaf turns blue-black, it proves that starch is present and it's produced by photosynthesis. Because remember, we first de-starched the plant. We removed all the starch. We checked also, when it was de-starched, the leaves were yellow-brown. When we started the experiment with the leaves and then we did another starch test, we saw that when we gave the leaves everything, starch was made, photosynthesis took place, and we removed that leaf and we did a starch test, it turned blue-black. So this is a positive starch test, and that's a negative starch test. What is the negative starch test? It remains yellow-brown. Positive starch test, it turns blue-black. Okay. So now here is an experiment, learners. This is where a plant was firstly de-starched, placed in a dark cupboard. Okay, all the starch was used up. Now the scientist wanted to investigate. I want to check whether light is necessary for photosynthesis. All right, so here's the leaf. Okay, here's the leaf. Now what they did was they de-starched the plant. They took it off the cupboard after 48 hours. Now the experiment is starting. They're now going to expose the leaves of that plant to light. Okay, so what are they going to do here? They're going to take one leaf and they're going to put a piece of foil. Okay, you're going to put a piece of foil. So here's the leaf, which is green. Now you're going to take a strip of foil, but before you put the foil up there, you're going to take a piece of foil. Remember, foil does not allow light to pass through. So you're going to take the foil and you're going to take a small blade and you're going to cut like a little cross through that foil like that. So here's the foil. All this is foil, foil, foil. And here is a cross. So that is the pattern of the foil, that it's cut through the foil so you can see through it. So you'll see that cross through it, through this foil, and that's the foil. Now you're going to take that piece of foil and place it over this leaf all around. So even the back of that will have the foil, and over here there's only that part that's got an opening that's going to allow light to pass through. So part A is exposed to light. That is exposed to light. Part B is the covered part of the foil. It's covered. It is the foil itself. So it's covering the leaf, no exposure to light. So it's covered, there's no exposure to light. Only that part is covered, is exposed to light, and that part and that part. So these are the parts that are exposed to light, and you need to know, or I must remember, that that's where my foil is. I can make a small mark there, that part never get light, you know it's there, and that part never get light, and all here and there, and this little cross here got light. Okay, so now it's got that pattern. So the diagram below shows you, let's just remove this here. Okay. So here's your experiment. Let's see now. The diagram below shows the leaf in a de-starched plant was set by a learner to perform an experiment. He wanted to see whether the presence of light will cause photosynthesis. So what was his aim? To check whether light is necessary for photosynthesis. So he first put the plant in a dark cupboard, de-starched it, then took it out. Now he's starting his experiment fresh. He took a piece of foil. He put it over the leaf. He cut the foil in the middle. We're making a cross. And then only those parts were exposed to light that was green. So initially the green parts are exposed to light, and the white parts, which represent the foil, is covering the leaf, preventing light from entering. So name the independent variable. What will be the variable that you are trying to control? Remember, an independent variable is the variable that the farmer or the learner or the scientist is using. He is controlling light. So the independent variable will be the presence of light, because that's what you want to investigate. So you must control that. You can't control how much of starch is going to be in the plant. So next one. What is the dependent variable? It's going to be the photosynthetic rate, but how are we going to know it? By the presence of starch. So the presence of starch depends on the amount of light. Am I right? So the light is what you are controlling, and you're going to measure it using the starch test. So the dependent variable will be the presence of starch. That is what you're going to measure. All right. Same experiment, different question. What is the aim of the investigation? Learners, always remember, the aim is always to say, to investigate whether light is necessary for photosynthesis, to investigate whether carbon dioxide is necessary for photosynthesis, okay? To investigate whether water is necessary for photosynthesis. So, in this case, we are talking about light. So the aim is going to be what? Okay, why is this not working? Okay. The aim is going to be to demonstrate or to investigate that light. Okay, what is the aim of this investigation? You can say to investigate whether light is necessary for photosynthesis, or you're doing this to show that light is necessary. Can you see that? You can use it in two ways. The aim could be to investigate whether light is necessary for photosynthesis, or you are doing this test to show someone that light is essential for photosynthesis. And how you're going to show that person is eventually by doing the starch test. So to demonstrate that light energy is necessary for photosynthesis. So how was the dependent variable measured? How was the presence of starch measured? How are you going to know whether it was there, that photosynthesis took place? You're going to test for the presence of starch. So the presence of starch is actually going to be tested in order for you to measure the rate of photosynthesis. Did photosynthesis take place? I got to test for the presence of starch, because my plant was de-starched. So if starch is there, it means photosynthesis took place. Now I must check where will photosynthesis take place. So now, learners, what you're going to do is, when you place this whole leaf now, after it's de-starched, for at least 24 hours in light, then on the third day now, or fourth day, you take this very same leaf. You must know exactly which parts were exposed. You boil it in the water, you let it get soft, then you take that leaf, you put it into a test tube, put ethanol, let it boil into the beak of water, remove that leaf, rinse it in the water, put it on the petri dish. It's now white, the leaf. Then you put iodine. So you're going to put a drop of iodine here where it was green. You're going to put a drop of iodine there where it was green. And you're going to put a drop of iodine where it was covered and where it was covered. And you're going to put a whole lot of iodine in this cross. And when you actually going to do the starch test, you're going to see that this leaf results. It's going to be, when the starch test, that all this here is going to be blue-black. All this is going to be blue-black. Even this is going to be blue-black, right? And then only here, it's going to be blue-black, because that's where starch will be formed, because these are parts exposed to light, even that little part there. But when you put a drop of iodine there and there, it's going to become yellow brown. When you put a drop of iodine here where it was covered and there where it's covered and all around here, it's going to be yellow brown, and only this cross here will turn blue black. So that means light is essential for photosynthesis, because the parts that were covered, not exposed to light, there was no starch, so no photosynthesis. And the part exposed to light turned blue black because photosynthesis took place and therefore starch was formed. And this is the conclusion. That a conclusion will be, according to the results of the starch test, light is necessary for photosynthesis. Remember, learners, your conclusion must always relate to the aim. Right. Same investigation again. The diagram shows that it was de-starched. The learner performed the experiment. He wanted to see if light will cause photosynthesis. What would be the color change observed in the areas of the leaf that were not exposed to light? That means this part here. It's going to be yellow-brown. Next question. What will be the color change in the areas of the parts that were exposed to light? That is here. It's going to be blue-black. Okay. Now here is another way you can get, again you can see, here's a strip of paper covering a foil strip covering the entire part of the middle part of the leaf. That part is exposed, and remember, you must first de-starch the plant before you start your experiment. You de-starch it, then you start your experiment. You cover it with the foil, you leave it in the sunlight, and then you can do the starch test. And you must know the areas that were exposed, because the leaf will be very white in all areas once you are boiling it in ethanol, right? So then when you place it in the petri dish, you must remember that that part was exposed to light and that part was exposed to light, and this middle piece was not exposed to light. And then you can see exactly where the starch is formed. So that is the entire starch test there. The results, the experiment, the leaf covered with a tin foil that was covered, no light. The iodine solution remained light brown, and in the control, the leaf that was left uncovered, the iodine solution turns blue-black. So that part's going to be blue-black, and that one's going to be yellow-brown. So they can always ask you, learners, to draw the results to show the color change in the leaf after the starch test. So this was before the starch test, right? De-starched, but the experiment, and now this is when you are doing the experiment. Now after the experiment, now you're testing for starch. They'll ask you to draw the results of the leaf after the starch test showing the presence of starch. So all you have to do is draw the very same leaf as it is drawn here with the petiole and things like that, right? And you got to draw the same leaf, and you're going to show the veins exactly like that, yeah? And then you're going to draw that, okay? Then you're going to show here that this part here is going to be blue-black, and that part there is going to be blue-black, and that this part here is going to be yellow-brown. And that is going to be what? You must always give your diagram a heading. Results of the results after the starch test. Results after the starch test. And what is this leaf here called? It's called the experiment leaf, because the factor that you're investigating is absent, but the control will have everything. So you can also have another leaf like that after you de-starch, where you don't put a foil and you put iodine everywhere, and the entire leaf will turn blue-black, saying that this one had everything, it had light everywhere. That's why there's starch everywhere. And where I covered it, there was no starch because there's no light. So this becomes my experiment. And when you gave it everything, that became my control. Okay. And what is the indicator that we use to test for starch? We use the iodine solution. What do we call it? We call it an indicator. All right. Conclusion, The parts that turn blue-black contain starch. They must ask you a question on the starch test. They must ask you a question on destarching the plant and the importance of destarching the plant before an experiment. You must know that, learners. The part which remains light brown does not contain starch. Therefore, the conclusion, light is essential for photosynthesis to take place and for the formation of starch or the formation of glucose and starch. But we say starch because all the glucose is joining. There's not just one small glucose. Many glucose molecules are forming during photosynthesis for the whole day. And then all of them join to form your starch granules, okay? So your conclusion is that light is essential for photosynthesis in order for the plant to form starch. And starch will be found where? In all your fruits and your vegetables, your beans, your carrots, your potatoes, everything, all food crops have starch because of photosynthesis. Now, learners, this is very important for you to know some facts about photosynthesis. And we speak about ATP, which we're also going to do in when you did respiration, you spoke about ATP. It's an energy carrier in cells. Also, this is very important for your next lesson on respiration. That ATP means adenosine triphosphate. And remember that ATP is an energy donor. It donates energy. Now, learners, remember that we spoke about photosynthesis were two phases. Ne? It was the light phase, which requires light. And light is necessary for what? Light is necessary for the energy to go into the chlorophyll. And then the chlorophyll energy, the energy of the light is going to energize the chlorophyll. And then that energy is going to be used to split water, which is called photolysis. And water, which is a molecule, is going to be split into hydrogen and oxygen. Remember that. Then that hydrogen that comes from the splitting of water in the light phase. You remember where does the light phase occur? It occurs in the grana, in the green parts, in the stacks of lamella where the chlorophyll is, that absorbs the light. Then the energy of the light activates the chlorophyll. Chlorophyll molecule's energy breaks down the water molecule into hydrogen and oxygen. Oxygen will be released during light phase as oxygen bubbles if you place it in water. But you can see you can't really see oxygen in the air, but if you put it into water, you can see the bubbles of oxygen. That means light phase is occurring. Then what happens to the H that comes from water? That's water. Oxygen will be released as O2. And this H is going to be accepted by something called a hydrogen acceptor. It's NADP, nicotinamide diphosphate. This one accepts the hydrogen. And this hydrogen will be used in the dark phase. So now while light was splitting water, energy was released in the form of ATP. So this ATP and this NADPH2, the hydrogen, the ATP from the light phase together with the H2 from the light phase will be used in the dark phase. And that is going to be used to form your starch. The dark phase does not mean it occurs in the dark. It simply means it's a phase in photosynthesis that does not require light. But it must occur immediately after the light phase. That question can come out. Why should the dark phase occur immediately after the light phase? Because the products of the light phase, hydrogen and ATP, will be used in the dark phase. Okay, so during photosynthesis, the energy from ATP is used in the reaction that makes energy-rich glucose molecules from the raw materials CO2 and water. So in which phase is glucose made? It's made in the dark phase. And for the dark phase, we need CO2, okay? And we need the hydrogen from the water. All right, and we need the energy from the ATP that came from the light phase. So ATP loses the phosphate and forms ADP. Sunlight can change the less active ADP into high energy ATP. So it's in the light phase whereby the light of the sun energizes the chlorophyll molecules, and then the energized electrons of the chlorophyll actually split water into hydrogen and oxygen. And when it does that, energy is released to ADP, which is the energy acceptor, to form ATP, and ATP and CO2 are going to be used, and hydrogen used in the dark phase to make your glucose molecules. Okay, so now you can see here's your ATP with 3 phosphates, adenine. There's your sugar, ATP, adenosine triphosphate. Adenosine, here's it, triphosphate means 3 molecules of phosphate. And what is ATP? It's an energy donor, and it's going to be used in the dark phase, whereby glucose is made. Learners also know, that the dark phase is also known as the Calvin cycle, because it was a person called Melvin Calvin. Melvin Calvin studied photosynthesis, and he discovered that it's in the dark phase, in the stroma of the chloroplast. That's the chloroplast. That's the grana. And in this stroma or matrix, that's where you had your RDP, which is your 5C compound, which joins with carbon dioxide from the air to form a 6C compound, which splits into 2 PGALs, phosphoglyceraldehydes. And then these phosphoglyceraldehydes use the hydrogen from the light phase, energy from the light phase, and then they're going to join to form your glucose-rich molecules, and all the glucose will join to form starch. That's in the dark phase. So remember, the dark phase is also called the Calvin cycle. And what do we need there? We need CO2 that's used, hydrogen from the light phase, and ATP from the light phase. That's why it occurs immediately after the light phase, because we need the product of the light phase to be used in the dark phase. It cannot be lying like that. It must be used immediately. And then that CO2 there will join with a chemical called RDP, ribulose diphosphate, inside the stroma, and that's called the Calvin cycle, where glucose is going to be made. And remember, it is in the light phase. What do we need there? We need which raw materials? We need water, and we need light, and we need chlorophyll. So the light phase needs light, and that light will be absorbed by the chlorophyll, and the energized electrons of the chlorophyll splits water into hydrogen and oxygen and releases ATP, and that ATP is going to be used in the dark phase, and that hydrogen coming from the water is also going to be used in the dark phase to make your C6H12O6. The C's are coming from the CO2, the O's are coming from the oxygen in CO2, and you find that water from the soil is giving you the H in your glucose. So your glucose, the hydrogens are coming from water, your O's are coming, your oxygen molecule there is coming from CO2, right? And your C's are coming from the carbon dioxide. And what else does photosynthesis give us besides making food? It makes for us oxygen. And that oxygen is coming from where? From the splitting of water. Can you see that? That's the oxygen that's making us to survive. You're breathing in oxygen. It's coming from photosynthesis. How did it happen? The splitting of water. What is it called? Photolysis. Which phase is oxygen made in? Light phase. Which phase are your fruits and your starches and your potatoes made in, and your fructose in grapes made in? The dark phase. What is the dark phase known as? Calvin cycle. Which raw material is used in the Calvin cycle? Carbon dioxide. Which two products of the light phase is used in the dark phase? Hydrogen and ATP. What is ATP? It is an energy donor. So children, you must make like a little schematic diagram, knowing that photosynthesis is a very valuable process. So the next time you see people cutting down trees and removing plants, tell them, what are you doing is you're actually killing the lungs of the earth. The lungs of the earth are the trees. Because without those trees, we cannot get food. Can you stay without food? No. So animals and humans cannot stay without food. Can you stay without oxygen? No. So the most important process of life is photosynthesis, because without that we cannot survive. And the sun's energy is the most valuable source of energy for all life on earth, because it's that energy, the radiant energy of the sun, that's trapped in the chlorophyll, gets converted into chemical potential energy that's stored in your glucose. And when you eat glucose and your enzymes break down the food during respiration in your cells, what is it going to release? ATP energy. And that's the energy you use to do work. So remember, learners, our next lesson is going to be on respiration, which means now, where we spoke about glucose being made during photosynthesis, respiration is how we take the glucose that's made from the food that we eat and we break it down to give us CO2 and energy. Okay, so what the plants give us is oxygen and food. Then we're going to take that food, break it down, and we're going to release CO2. So that's all from me today, learners. Please study your work, because the light phase, the dark phase, the destarching, the experiments will all be tested in the June exams. Thank you so much.